I want you to imagine a heavily guarded fortress, like we're talking thick, reinforced concrete walls.
Oh, yeah.
Heavily armed patrols, and, a security system that is just completely impenetrable.
Right, a total lockdown.
Exactly.
Okay.
Because for decades, this is exactly how oncologists have viewed the tumor microenvironment of pancreatic ductal adenocarcinoma, or PDAC.
Yeah, it's notoriously stubborn.
It really is one of the most difficult cancers to treat. And the elite guards of this fortress, they're this type of immune cell called regulatory T cells.
Or Treg cells, yeah.
Right, Treg cells. And their entire job is to actively suppress the body's natural immune response, right? They just patrol the walls of this tumor and completely shut down any rogue immune cells that are, trying to mount an attack.
That's their whole biological purpose in that space.
But what if, instead of just trying to bomb the fortress or, I don't know, kill those guards, you could somehow brainwash them? Like literally brainwash them into turning on the fortress itself.
I mean, it sounds a bit like a sci-fi movie plot, right? But that concept is exactly the premise of the research we are diving into today. It represents a massive paradigm shift in how we approach tumor immunology. We aren't just fighting the tumor anymore. We're actively trying to rewrite the loyalties of the cells protecting it. Man,
that is so cool. So our mission for this deep dive is to unpack a truly fascinating 2026 article from the journal Immunity by Maltese and colleagues.
Yep, great paper.
We're looking at a huge stack of source material today. I mean, highly detailed immunological data, complex spatial imaging maps, and actual clinical trial results.
The real deal human data.
Exactly. And together, they prove that we can actually reprogram these immune-suppressing cells right inside the tumor.
Okay, let's unpack this. But before we get into the cellular brainwashing, we need to understand the baseline.
Right, the current standard of care.
Yeah. What is the standard approach to immunotherapy, and, why does it hit a brick wall when it comes to pancreatic cancer? So to understand the baseline, we have to look at immune checkpoint blockades. If you follow oncology even casually, you've likely heard of drugs called anti-PD-1 or anti-CTLA-4.
Oh, yeah, those are huge right now.
They are. And the classic analogy is that these drugs basically take the brakes off the immune system. You see, a tumor usually tricks the immune system into halting its attack by engaging these biological brakes.
I need.
Very. So checkpoint inhibitors block that interaction. Theoretically, they take the brakes off, allowing your immune cells to rev up and just destroy the cancer.
Right, so you take the brakes off, the immune system speeds up, runs over the tumor. Sounds great.
It does sound great. But here's the critical flaw in that approach when it comes to pancreatic ductal adenocarcinoma. Taking the brakes off a car doesn't do you any good if that car is literally buried underneath a concrete bunker.
Ah, I see.
The microenvironment of a pancreatic tumor is incredibly dense and just profoundly suppressive. Those checkpoint blockade drugs often fail because there are simply too many suppressive signals actively holding the immune system down.
And that suppression is heavily driven by those elite guards you mentioned, the Treg cells.
Exactly. So the new experimental approach detailed in this paper introduces a totally different kind of tool. It's an agonistic anti-CD40 antibody, often referred to as alpha-CD40.
Wait, agonistic, like meaning it stimulates rather than blocks.
Precisely. A blocking antibody stops a signal from happening. But an agonistic antibody acts like a chemical duplicate. It binds to a receptor and actively pulls the trigger to start a signal.
Oh, wow.
Yeah. Instead of just taking the brakes off, an agonist physically steps on the gas pedal of the immune system.
Okay, so we have the fortress, we have the guards, the Treg cells, and now we are introducing this new weapon, the alpha-CD40 agonist.
Yeah.
Let's look closer at these guards. What makes a Treg cell so stubbornly protective of the tumor?
Well, regulatory T cells are a highly stable subset of CD4 positive T cells. Now, to be clear, in a healthy human body, their job is absolutely essential.
Right, they aren't always the bad guys.
No, not at all. Imagine you get a severe viral infection. Your immune system goes to war, causing massive inflammation. Once the virus is dead, you need a mechanism to calm the immune system down, right? So it doesn't start destroying your own healthy tissue.
Because that would be an autoimmune disease.
Exactly. That's what Tregs do. They prevent autoimmune diseases. But in a tumor microenvironment, they are essentially hijacked by the cancer. Their entire identity, their suppressive nature, is completely locked in by a master transcription factor called FOXP3.
FOXP3. Okay, I like to think of a master transcription factor as the cell's underlying operating system, or, maybe its security badge.
I like the badge analogy.
Yeah, like if a cell holds that FOXP3 badge, Its absolute biological mandate is to suppress the immune system and protect whatever environment it's in, which, in this case, is the tumor.
That's a really great way to visualize it. And this brings us to the first major observation of the study. When the researchers treated these pancreatic tumors in mice with the alpha-CD40 drug alongside standard checkpoint blockades, the number of these suppressive FOXP3 positive Treg cells absolutely plummeted.
Just wiped out.
A total collapse of the guard force. And it happened incredibly fast, like within just 48 hours.
Hold on a second. I'm thinking through the mechanics of this and looking at the notes. There is a massive scientific paradox here.
Yep, you caught it.
The source material states very clearly that Treg cells do not express CD40 receptors on their surface. So how on earth is this alpha CD40 drug killing a cell it literally cannot touch?
It's a great question.
Because antibodies work like a lock and key, right?
Yeah.
If the Treg doesn't have the CD40 lock, the drug can't do anything to it. That's like trying to open a steel door with a radio wave.
What's fascinating here is that you've hit on the exact core mystery that drove this entire paper. The researchers saw the Tregs disappearing, but they knew the alpha CD40 drug was physically incapable of binding to them.
Right. Therefore, the drug must be using a middleman. It has to be talking to a completely different cell in the tumor microenvironment, and that cell must be the one ordering the hit on the Tregs.
Okay, so let me just recap this for everyone listening before we move on. So PDA tumors use these FOXP3 Treg cells to suppress the immune system.
Correct.
We inject an alpha CD40 drug, and the Tregs suddenly disappear in 48 hours. But because the drug can't touch the Tregs, there's a ghost in the machine.
A hidden variable.
Yeah. So if you're listening to this, you're probably wondering the exact same thing the scientists were. Who exactly is the drug talking to in the tumor microenvironment to order this hit?
So to find the middleman, the researchers had to map out which cells in the tumor actually do express that specific CD40 receptor. And the primary targets turned out to be a type of antigen-presenting cell known as conventional type 1 dendritic cells, or CDC1s.
Dendritic cells. I've always thought of them as the sentinels or the scouts of the immune system.
That's spot.
They usually gather intel and show it to the T cells. Right. Exactly. And they are heavily armed with CD40 receptors. So the researchers mapped out the mechanism, and it works like a chemical chain reaction. Step 1, the alpha CD40 drug enters the tumor and binds directly to the CD40 receptors on those CDC1 dendritic cells.
Okay.
This aggressively stimulates them. Step 2, these activated dendritic cells essentially panic and release a powerful chemical alarm. Specifically, they pump out a cytokine known as interleukin-12 or IL-12.
IL-12, got it.
And in step 3, that IL-12 acts as a catalyst, triggering a massive secondary production of interferon-gamma. And interferon-gamma is a fiercely anti-tumor, highly inflammatory signal.
Okay, I can visualize this now. Returning to our fortress analogy. The dendritic cells are the radio operators sitting inside the base.
I love this.
The alpha CD40 drug is a direct command from the general coming in from the outside.
Right.
The radio operators receive the command, and they immediately blast interleukin-12 and interferon-gamma. Those cytokines are the radio frequencies broadcasting the attack order across the entire battlefield.
That is exactly right. But in molecular biology, simply proposing a logical mechanism isn't enough. You have to prove it.
Right, you need evidence.
And the way you prove a mechanism is by deliberately breaking it, piece by piece, to see if the whole system fails. The researchers did this brilliantly using genetically modified knockout mice.
Okay, wait. Knockout mice, meaning they literally edit the DNA of the mice to, like, knock out or remove a specific gene. So the mouse is born missing one very specific biological component.
Precisely. It's a very target deletion. They tested the alpha CD40 drug on 3 different variations of knockout mice.
Right.
First, they used BATF3 knockout mice.
That's 3.
Yeah, without the BATF3 gene, these mice completely lack those specific CDC1 dendritic cells. They essentially remove the radio operators.
Okay, that's one link gone.
Second, they used IL-12P40 knockout mice. This means the mice cannot physically produce the interleukin-12 cytokine. So they removed the first radio frequency.
Wow.
And third, they used interferon-gamma knockout mice, completely removing the downstream inflammatory signal.
Let me guess. Without the radio operators, or without either of those specific radio signals, the drug did absolutely nothing.
It completely failed. In all 3 of those genetically modified scenarios, the alpha CD40 drug had 0 effect on the Tregs.
0.
0. The suppressive guards stayed right where they were, protecting the tumor. By breaking the chain at 3 different links, the researchers proved definitively that this specific IL-12 to interferon gamma cytokine axis is absolutely mandatory to clear out the Tregs.
Man, that is just incredibly elegant science. If you pull out even one piece of the communication chain, the whole attack just falls apart.
It's beautiful, really.
So just to do another quick recap for you listening, the alpha CD40 drug activates the CDC1 dendritic cells. Those cells blast IL-12, which drives interferon gamma. And this whole cytokine axis is required to eliminate the Tregs.
That's the chain.
But this brings us to a really crucial, almost deceptive twist in the research. We know the radio signal goes out and the Tregs disappear. But if you're paying close attention, you're probably asking, did they actually die or did they do something much more deceptive?
This raises such an important question, and it's exactly what the scientific community initially assumed. The standard thought process was apoptosis.
Meaning cellular suicide.
Right. Everyone thought that the intense interferon gamma signaling was simply toxic to the Tregs, causing them to self-destruct. But to be absolutely sure, the researchers utilized a technique called FOXP3 lineage tracing.
Okay.
And the results from this technique are, honestly, arguably the most mind-bending part of the entire paper.
Lineage tracing. I'm looking through the methodology here, and it involves a drug called tamoxifen and some sort of fluorescent dye. Can you break down how scientists actually track the lineage of a microscopic cell in a living animal?
Yeah, think of it as a permanent molecular tracking device. The scientists engineered the mice with a very specific genetic trigger. When they inject these mice with a drug called tamoxifen, it acts like genetic scissors.
Scissors.
Yes, it cuts a piece of DNA in any cell that is currently expressing that FOXP3 master transcription factor. When that cut is made, it permanently turns on a gene that produces a glowing red fluorescent protein called TD tomato.
PD tomato, that's a great name. So if a cell is a suppressive Treg holding that FOXP3 badge at the exact moment they inject the tamoxifen, it basically lights up bright red under a microscope.
Yes. But here is the absolute genius of lineage tracing. Because the tamoxifen literally edited the cell's DNA to turn on that red dye, the red tag is permanent.
Wait, permanent?
Yes. It does not matter what the cell does tomorrow. Even if the cell completely stops making FOXP3, even if it completely changes its identity, it will still glow red forever. It's a permanent historical marker of what the cell used to be.
Oh, wow. Okay, here's where it gets really interesting for anyone listening. The researchers give the alpha CD40 drug. They wait 48 hours. They look into the tumor. The FOXP3 badges are gone, which we knew. But when they look for the glowing red PD tomato dye,
the red cells are still there.
They're still there. The overall number of red glowing cells inside the tumor hadn't dropped at all.
Exactly. The Tregs didn't undergo apoptosis. They didn't die. They didn't pack up and flee the tumor to the lymph nodes. They stayed right there on the battlefield.
That's crazy.
But they physically lost their FOXP3 expression. They literally threw away their suppressive security patches. We now refer to these converted cells as ExTreg cells.
ExTregs, like former employees of the tumor fortress. But if they're no longer suppressing the immune system, what are they actually doing? I mean, they didn't just quit their jobs and sit around playing cards, did they?
They absolutely did not. And we can see exactly what they became by looking at the other transcription factors inside them. Under the heavy influence of that interferon gamma radio signal we talked about earlier, these ExTregs downregulated a protein called Helios.
Helios, what does that do?
Helios normally acts like an anchor, stabilizing their suppressive protective nature. By dropping Helios, they unmoored themselves. But simultaneously, they massively upregulated a completely different master transcription factor called TBET.
TBET. Now, why is that specific transcription factor so important to this story?
Because TBET is the master transcription factor that drives Th1 effector cells. Th1 cells are the aggressive, cancer-killing attackers of the immune system.
Oh, so it's a totally different identity.
Completely. In the world of cellular biology, FOXP3 and TBET are diametrically opposed. They are enemies. By losing FOXP3 and gaining TBET, these cells aren't just retiring, they are literally putting on the armor of the attacking force.
Wait, putting on the armor is one thing, but are they actually fighting? Like, how do we know they aren't just wearing the uniform for show?
The researchers proved they were fighting by using a highly sensitive technique called RNA-scope. It allows scientists to look inside the cell and see exactly which RNA transcripts are currently being manufactured.
RNA being the blueprints the cell is actively reading to build proteins.
Exactly. And the RNA-scope revealed that these ExTregs were actively manufacturing their own interferon gamma.
No way!
Yes. Think about what that means. They weren't just passively listening to the inflammatory radio signal anymore. They had become active broadcasters of that exact same attack signal.
That is mind-bending.
They completely fundamentally flipped allegiances, from immune suppressors dedicated to protecting the tumor to active immune attackers fighting to destroy it.
So let's recap this massive twist for everyone. Lineage tracing proved the Tregs do not die after the alpha CD40 treatment. They change identity, they drop the FOXP3 badge and become ExTregs.
Yeah.
They pick up the TDET marker and actually start making their own anti-tumor interferon gamma. So you have these newly brainwashed soldiers, these ExTregs, inside the tumor. But my question for you listening is, where exactly do they go to fight? Because a tumor is a physical three-dimensional structure.
Right. It's not just a soup of cells. To figure out the spatial dynamics, the researchers had to build a literal map. They used an advanced, highly multiplexed tissue imaging technology called IBE.
IBEX. Multiplex meaning they can see a lot of things at once.
Exactly. Standard imaging might let you see 3 or 4 different proteins at once. IBEX allows researchers to stain for dozens of different proteins simultaneously on a single slice of tissue.
Wow, that's incredibly detailed.
It creates a hyper-detailed physical map of the cellular battlefield. And what they found was that these ExTregs were not just floating around randomly, they physically migrated outward, heavily concentrating at the very outer border or the periphery of the tumor.
The border. Why the border? Are they like trying to escape the tumor microenvironment entirely?
Not at all. They are moving there with purpose, following a chemical trail. Remember those CDC1 dendritic cells we talked about, the original radio operators?
Yeah.
It turns out they are also heavily positioned at the tumor border. And these dendritic cells release a very specific chemical attractant, a chemokine, called CXCL9.
And let me guess. The ExTregs happen to have the exact receptor needed to detect that chemical.
You got it. A receptor called CXCR3. If we connect this to the bigger picture, it acts like a biological magnet. The dendritic cells at the border are releasing CXCL9, and the CXCR3 receptors on the ExTregs pull them directly to the front lines where the dendritic cells are clustered.
It's like a highly coordinated, intentional spatial reorganization of the whole immune landscape.
It really is.
Okay, so the ExTregs migrate to the border, and they hang out with the dendritic cells. But, you know, just because you're standing on the front line doesn't mean you're actually firing your weapon. How did the scientists prove that these reprogrammed cells were actively engaged in combat with the cancer?
They proved it using a brilliant, highly specific metric. They measured the physical location of a protein called NFAT1.
NFAT1. Unpack that for us.
When a T cell is just resting, not really doing anything, the NFAT1 protein floats around in the cell's cytoplasm, which is the jelly-like substance outside the nucleus.
Right.
But the exact literal moment the T cell's receptor specifically recognizes a tumor antigen, meaning it has found a target and is actively engaging it, that NFAT1 protein physically moves from the cytoplasm directly inside the cell's nucleus to help turn on activation genes.
So finding NFAT1 inside the nucleus is essentially catching the cell red-handed, pulling the trigger.
Precisely. It is unequivocal proof of target engagement. And when their researchers looked at their spatial maps, they found that the ExTregs sitting at the tumor border had the highest levels of nuclear NFAT1 out of all the T cells in the entire tumor.
Wait, really? The highest?
The absolute highest. In fact, their engagement levels were higher than the standard CD8 killer T cells, which are usually the primary attackers. The ExTregs weren't bystanders. They were the most active, fiercely engaged cells on the entire battlefield.
That is absolutely staggering. We've taken a cell whose sole biological purpose was to protect the cancer and turn it into the cancer's most aggressive enemy.
It's a complete reversal.
Okay, quick recap before the final leg of our journey. The ExTregs migrate to the tumor periphery alongside the dendritic cells, guided by the CXCL9 magnet. And nuclear NFAT1 imaging proves they are actively fighting. But for you listening, you're probably asking the ultimate question right now. This is all incredible in mice, but does this cellular brainwashing actually happen in human beings with pancreatic cancer?
It's the most critical question in any translational research because, curing cancer in a mouse is one thing. Doing it in a human is entirely different. But the authors of this source material didn't stop at animal models. They took this mechanism directly to the clinic. They analyzed resected pancreatic tumors from human patients who were enrolled in a phase I clinical trial.
A phase I trial. So this is early stage testing in humans, primarily looking at safety, but also looking for early signs that the drug actually works biologically.
Exactly.
I saw in the notes that these were patients with resectable pancreatic cancer, meaning the tumor could be surgically removed, and they were given a neoadjuvant alpha CD40 drug called celicrilimab. What exactly does neoadjuvant mean in this context?
Neoadjuvant simply means the therapy is given before the main treatment, which in this case is surgery. They gave the patients the CD40 agonist roughly 12 days before their scheduled surgery.
Okay, so a 2-week head start.
Right. This is a crucial detail because it gave the drug a nearly 2-week window to alter the tumor microenvironment while it was still inside the patient's body. Then, when the surgeons removed the tumor, the researchers had a perfectly preserved snapshot of what the drug had done.
And they analyzed these human tumors using multiplexed immunohistochemistry, which is essentially the human pathology equivalent of that IBEX mapping we discussed earlier.
Yep, same idea.
They painted these specific proteins with microscopic dyes to see exactly where different cells were living.
Yeah.
So, how did the human tumors compare to the mice?
The results beautifully mirrored the animal models. When they compared the tumors of patients who received the alpha CD40 drug against tumors from patients who went straight to surgery without the drug, the difference was totally clear.
What did they see?
In the treated patients, they saw a distinct accumulation of T cells that were positive for both FOXP3 and T-bet.
Oh, wow. So they caught the human cells mid-transition, wearing the attacker armor.
Exactly. And just like in the spatial maps from the mice, these Th1-skewed, reprogrammed Tregs were heavily concentrated at the outer border of the tumor, physically clustered right next to the dendritic cells.
Wow. So let's wrap this up for the listener. Our final summary bullet points. Clinical trial data confirms this same process happens in human PDAC. Treated patients show T-bet positive Tregs at the tumor border. So what does this all mean for the listener? Why should they care about the exact positioning of a reprogrammed cell in a pathology slide?
Well, it means everything because it proves that the underlying biological mechanism is conserved across species. The ability to take a highly stable bad actor cell and force it to switch sides via type 1 inflammation isn't just a quirk of mouse genetics.
It's universal.
It is a fundamental rule of mammalian biology. It works in mice, and the clinical data strongly suggests the exact same reprogramming happens in human patients.
Which brings us perfectly to the end of our deep dive today. Let's take a step back and look at the whole picture. We started with an impenetrable fortress, a pancreatic tumor heavily guarded by an elite force of suppressive Tregs.
Yep, the locked-in guards.
For years, we thought the only way to win was to find a drug that could selectively kill those guards. But this research showed us a completely different path. By introducing an agonistic CD40 antibody, we didn't drop a bomb on the guards. We handed a direct order to the dendritic cells.
Our radio operators.
Exactly. Those dendritic cells broadcast a massive chemical alarm made of IL-12 and interferon gamma. And that alarm was so powerful that it actually reached into the DNA of the Tregs, forcing them to strip off their suppressive FOXP3 badges, pick up T-bet swords, and transform into ExTregs, fighting fiercely at the very edge of the tumor.
It is a strategy of subversion rather than mere destruction, but, beyond just pancreatic cancer, it forces us to completely rethink the permanence of cellular identity.
What do you mean?
In biology, we are often taught that once a cell differentiates, once it gets that FOXP3 badge, its destiny is permanently locked. We're taught that a bad cell is a bad cell. But this deep dive proves that cellular identity is highly plastic. It can be dynamically rewritten directly inside the living body, simply by exposing it to the exact right combination of chemical whispers.
And that leaves us with one final provocative thought for you to ponder as you go about your day. We've just seen proof that a highly stable tumor-protecting immune cell can be reprogrammed into a tumor-killing attacker just by tweaking the chemical environment. If that's possible, what other bad actor cells in the human body are just sitting there waiting to be flipped?
It's a huge question.
Think about the aggressive immune cells driving autoimmune diseases, or the exhausted cells failing to fight off chronic infections, or even the cellular process of aging itself. How many of those stubborn locked-in cells aren't actually permanently broken, but are just waiting for us to figure out the exact biological radio frequency needed to make them switch sides?